Why are ESAT-6 and CFP-10 important for IGRA-TB tests? Have you ever wondered why certain proteins are crucial for diagnosing tuberculosis? Let's dive into the importance of ESAT-6 and CFP-10 in Interferon-Gamma Release Assays, or IGRAs, which are used to detect latent tuberculosis infection. ESAT-6, which stands for Early Secreted Antigenic Target 6 KDA, and CFP-10, or culture filtrate protein 10 KDA, are specific proteins secreted by the bacteria Mycobacterium tuberculosis. These proteins are encoded in a part of the MTB genome that is not present in the Bacillus calmette-guerin vaccine strain or in most non-tuberculous mycobacteria. This unique absence makes ESAT-6 and CFP-10 reliable markers for identifying MTB infection. When you undergo an IGRA test, your immune system's response to these proteins is measured. If you have been infected with MTB, your immune cells will recognize ESAT-6 and CFP-10 and produce a substance called interferon-gamma. The test detects this interferon-gamma releases, which indicates a positive result for latent TB infection. This method is more specific than the traditional tuberculin skin test, which can sometimes give false positives due to prior BCG vaccination or exposure to environmental mycobacteria. The combination of ESAT-6 and CFP-10 in IGRA tests is particularly important. Using both proteins together enhances the test's sensitivity and specificity. For instance, newer versions of IGRA, like QuantiFERON-TB Gold Plus, include peptides from both ESAT-6 and CFP-10 to stimulate different types of immune cells, improving detection accuracy. However, it is essential to note that there are some limitations. Certain non-tuberculous mycobacteria may have genes similar to ESAT-6 and CFP-10, which could lead to cross-reactivity and potentially false results, though this is relatively rare. Additionally, since ESAT-6 is also being studied for TB vaccine development, interpreting IGRA results in vaccinated individuals can be complex. This has led to ongoing research into alternatives that do not include ESAT-6. In summary, ESAT-6 and CFP-10 are vital for IGRA-TB tests because they serve as specific immune targets unique to MTB infection. This specificity helps accurately identify latent TB infection while reducing false positives related to BCG vaccination or exposure to environmental mycobacteria. Their role is central to current TB screening strategies using IGRA technology. If you have questions about TB testing or your health, it is always best to consult with a healthcare professional for personalized advice.